Hi, my name is Chante Bethell, and I am a biological data analyst at the Childhood Cancer Data Lab powered by Alex's Lemonade Stand Foundation. And today I'm going to talk to you about a single sample predictor project for medulloblastoma subtypes. Medulloblastoma, shorthand MB, is an aggressive cancer of the cerebellum with 4 subtypes that have unique molecular features and patient outcomes. The subtypes WNT, SHH, Group 3, and Group 4 we know are well characterized in large cohort studies involving transcriptomic and epigenomic data. Although Group 3 and Group 4 are notably difficult to distinguish from transcriptomic data. Less development, however, has been made in predictors that can be applied to any individual sample at any given time. These single sample predictors are nice because they can accurately classify individual samples without normalizing to match a training distribution, which is unlike multi-sample predictors, which expect a normalized data set before training and prediction can occur. Single sample predictors are theoretically nice as well because training and compute time for these single samples would be a lot less than for large cohorts. But before we go deeper into single sample predictors, let's look at a multi-sample predictor and how that would work. So for multi-sample predictors, you have a training data set with subtype labels that's used to produce a classification model. And that classification model is then applied to a cohort of gene expression data. And predicted subtype labels are then the results or the output of this classification model being applied to that data set. So you get subtype labels for that entire cohort of data, whereas with a single sample predictor, training is fairly similar. You do have a training data set with subtype labels that's used to produce that classification model. However, for prediction, you can then apply that classification model to an individual sample gene expression data and get the predicted subtype label on an individual sample basis. So at any given time, you can apply this model to a sample and get that predicted subtype label. So our model uses transcriptomic data. If you take a look at the plot on the left, you'll see that we have a variety of array and RNA-seq medulloblastoma data. Although we do have more array data, you'll see that the proportion across array and RNA-seq is comparable across subtypes. So we train on training data and we apply to the test data. For array and RNA-seq, we choose 50% of studies to go into training and all other studies into testing. And this splitting of training and testing is repeated 10 times. And out of 10, the best performing model is applied to the validation set. So we perform classification using k-top scoring pairs and random forests. And that's because these methods have relative expression rules like gene A is less than gene B, which can be implemented for a robust classification. So during training, each rule is indicative of a subtype. Let's start by looking at subtype G3 or group 3. If the rule is true, that subtype gets an upvote. So here you'll see for group 3, you have a saturated square that almost looks fully black. Each black tick in this square represents an upvote for that particular rule. If we look at the remaining subtypes, you'll see that there's less upticks that's associated with these subtypes in this particular column that's referencing group 3, as expected. These upvotes or upticks contribute to the scores. And again, you'll see that the scores for group 3 are much more saturated than for group 4, SHH, and WNT. And the same will be true for the remaining of the subtypes in the dataset. Random forest also uses these relative expression rules to make decision trees, making these classification methods promising for our purpose. The R package, multi-class pairs, implements a KTSB and random forest approach as well, so we're able to use the functions from this R library for testing in our project. So we found that the KTSB and random forest models perform well across platforms. We use Kappa to assess the performance. Kappa is a measure of performance that takes distribution of classes into account. And we also decided to compare these models to 2 other classification methods, one being lasso that we use as our baseline method. It's a comparative method because it's often used in studies. And we are also looking at MM2S, which is a method specifically designed for medulloblastoma subtype prediction. You see that the values are highest and most consistent across repeats for the KTSP and the random forest models. However, they are comparable to the lasso model although the lasso model appears to have an outlier that is skewing that RNA-seq data. KTSP and random forest models also outperform these existing prediction methods when we look at the specific subtypes. So we're looking at group 3 and group 4, and here we are using balanced accuracy to assess these methods. And you'll see that again, KTSP and random forest remain high and consistent and comparable to lasso, who also still has that outlier shown here, skewing that RNA-seq data. Now, can these pair-based methods use fewer genes as input for prediction? We know that rules learned by the pair-based KTSP and random forest models don't use all genes in the transcriptome, and not all samples assayed for gene expression will use transcriptome-wide platforms. So to test performance in the context of targeted assays, we use a collection of reduced genes on a pan-cancer nanostring panel to train new models. We found that the models trained on the reduced gene set performed similarly well to the transcriptome-wide models. KTSP and random forest again the performance remained consistent and high across repeats. It appears that KTSP and random forest are flexible to handle different gene sets, whereas lasso and MM2S do not work as well because there's access to a different set of genes while training the model, and now the test data set has a totally different set of genes. Can these KTSP and random forest models now be applied to single-cell RNA-seq data? Can we use the models that are trained on bulk data to predict subtype when we only have single-cell RNA-seq data available for a particular tumor? We tested this by creating pseudobulk data. We merged expression from all cells in 36 single-cell samples, and we applied the models that were trained on bulk data to this pseudobulk data set. And we found that the KTSP model performs well on this pseudobulk single-cell data set. Most of the pseudobulk samples were confidently predicted as their true subtype label. So if we take a look at the plot on the right, on the x-axis, we have score percentage, which is going to be the percentage of the prediction scores that were signaled or provided for each sample. So lasso and MM2S cannot make predictions in the settings because, yet again, there were genes in the model that are not present in the pseudobulk data. And we did find 3 samples that show a mix of G3 and G4 in this case. So if you take a look at the first sample here, you'll see that it is hovering around that 50% mark. So there seems to be a very nice mix of G3 and G4 in this G3 sample. Again, in the G3 panel or facet, you'll see something similar in this sample. And in the G4 panel, you'll see something similar in this sample. Now we want to look at, can KTSP and random forest models be applied to individual cells? We know that expression data for individual cells is sparse. Not all genes are going to be detected. An advantage of these KTSP and random forest models is that fewer genes are used for prediction. Group 3 and Group 4 have also been found to exist as intermediates on a transcriptomic spectrum. Obtaining these subtype labels for individual cells may help us explore this idea even further. So we applied these KTSP and random forest models that were trained on bulk data to individual single cells. And we found that most individual cells are predicted as a subtype label, while some samples do show a mix of, yet again, Group 3 and Group 4. So this sample is actually one of the samples that were highlighted earlier. You can see that there is a nice mixture of yellow and blue here that signify Group 3 and Group 4. It does appear that on this UMAP, there may be some clustering starting to happen, but it definitely is a nice mix of the phenotypes Group 3 and Group 4. Our single-cell prediction confidence scores also show this mix of Group 3 and Group 4 in those 3 samples that were highlighted earlier. In this case, confidence is the reliability of the subtype prediction, and the dotted line at about 75% represents that 75% of the prediction score matches this subtype. So if we take a look at this first panel, for example, you'll see that Group 3 and Group 4, they are quite comparable when it comes to their confidence scores across repeats. And the same is happening in the other panels. So there seems to be a very nice mix of group 3 and group 4 in these individual samples. So overall, we've determined that single sample predictors do work with bulk and single-cell RNA-seq data. Notably, KTSP and random forest perform well across subtypes, across platforms, and for bulk and single-cell samples. The predictions for those 3 samples in the single-cell experiments that were a mix of group 3 and group 4 do support a model of group 3 and group 4 existing along a continuum, and also illustrate the value of the ability to classify individual cells. And a future direction for us may involve extending these pair-based methods to include epigenomic data for multi-omic studies. Thank you for having me, and thank you to the authors, Stephen Foltz, who is leading this project, as well as Casey Grain and Jacqueline Taroni. And if you'd like to find more about this project, please visit our GitHub and visit the medulloblastoma classifier repo.